Okay, so this is in your packet of everything that's going to be covered in the lecture. I'm not going to read it to you because you can read it for yourself. And this is just like an idea of everything that we're going to be talking about today. So please make sure you read that so that you know what's coming up. So to begin with, enzyme kinetics is the focus of our second experiment in this class. And enzyme kinetics is the study of rates of enzyme-catalyzed reactions and how they're changing in response to experimental conditions. So when people are interested in enzymes, they are looking at the rates of a reaction with changing substrate concentrations or changing the pH of the solution, or if there's a particular inhibitor that might be present in solution or in a cell. If you're talking about the human body, they're looking at the overall picture of how we can look at the particular rate of a reaction and how maybe an inhibitor or something that's going to affect an effector is going to make the reaction run better, how they're affecting that rate of the reaction. So the study of enzyme kinetics is necessary for determining the binding affinities of substrates and effectors to an enzyme and understanding the reaction mechanism. And they're doing this so that they can understand the role of an enzyme in the metabolic pathway, which then in turn leads to being able to develop reagents for biochemical analysis or developing useful drugs. One example of this is in HIV. People who had AIDS in early days were just dying until they were able to develop specific drugs which were going to inhibit some of the proteins that the virus was using to be fully active. And as long as people keep taking these specific inhibitory drugs, they can keep their HIV in check. So this is a real-world example of how enzyme kinetics was applied to a situation to directly help human survival. So when we're talking about enzymes, remember from your basic biochemistry that enzymes are simply usually protein catalysts. A larger picture of enzymes has sometimes RNA and things like that in there, but at its basic level, a protein catalyst. And this catalyst is going to take a substrate that's going to turn into a product. But remember from your basic chemistry that a catalyst is simply going to speed up a reaction. If it's not energetically favorable, a catalyst will not be able to have an effect on the particular reaction that you're looking at. So it's going to turn a substrate into a product, and it's going to speed up the biochemical reaction 10 to the 6 to 10 to the 17 times. So that's all a catalyst is going to do, is either hold the substrate and the substrates, together in an energetically favorable conformation to allow them time to react. It can do various things like that to help the reaction occur a little bit faster. So the specific reaction that we are talking about in this upcoming experiment is the lactate dehydrogenase reaction. And the lactate dehydrogenase reaction takes pyruvate and reacts it with NADH in the presence of an extra hydrogen. And that lactate dehydrogenase takes that pyruvate and the NADH and turns it into lactate. Basically, across the carbonyl carbon right here, it adds 2 hydrogens across that carbonyl carbon. 1 hydrogen that it gets from the solution, 1 hydrogen that it's getting from NADH, and so it's turning it into lactate plus NAD. So that's the entire reaction right there. Now, the substrates for this reaction are pyruvate and NADH. NADH is also sometimes called a cofactor for the reaction. And when you add LDH to the reaction, the concentration of pyruvate is going to decrease. The concentration of NADH is going to decrease. The concentration of lactate is going to increase because that's a product of the reaction. and the concentration of NAD is going to increase because that's where the hydrogen is coming, because the NADH is turning into NAD after that hydrogen is removed. So there's a little bit of math that governs this idea, and it's called Michaelis-Menten kinetics. So Michaelis-Menten kinetics is basically the idea that there is a steady state for the reaction, that it's governed by a particular step of the reaction. So in this case, we have the enzyme plus substrate, and there's going to be a particular rate that enzyme plus substrate is going to be reacting into an enzyme substrate complex, and then that enzyme substrate complex is going to decay. And there's two different ways that enzyme substrate complex can decay. It can either go to the original enzyme plus product, and that's governed by a particular rate, or it can go back to enzyme substrate can degrade back into enzyme plus substrate separately. And so each of those have specific rates associated with them, and there's going to be a rate that's the slowest rate of the reaction. So very often it's the enzyme substrate complex making the product and regenerating the original enzyme that is the slowest rate. And so it's called steady state because these can be happening very fast, but that's governing the rate that the reaction is happening. So the rate of enzyme substrate formation is K1, the rate that they can combine, times the enzyme and the concentration of the enzyme and the concentration of the substrate. So the higher the enzyme or the higher the substrate, the faster that reaction can be happening. The rate of the enzyme substrate breakdown, there's two different ways. Remember that it can break down. It can either go to product or it can either go back to enzyme plus substrate after the ES complex is formed. So that's K minus 1, the back rate, times the enzyme substrate complex, plus K2 times the enzyme substrate complex. So both ways in which it can break down. So when you have what's called steady state kinetics, it's saying that the rate of formation is going to equal the rate of breakdown. So K1 times the enzyme times the substrate is going to, so that's the rate of the formation, is going to equal the rate of the breakdown. We're not going to get too much into the math in this class, just a very, very tiny little bit. But basically what comes out of this idea is the velocity of the reaction is the catalytic rate, the K2, times the enzyme substrate complex. And so the maximal velocity is that catalytic rate times the total amount of enzyme. So this is an important equation right here. Vmax equals K2 or Kcat times the total amount of enzyme. And the Km of the reaction, so the Km is the dissociation constant for that enzyme substrate complex. So that's going to equal K minus 1 plus K2 over K1. Now, when a bunch of math is done on this idea, we end up with the Michaelis-Menten equation. So this is another equation that you're going to be expected to know for this class. So the Michaelis-Menten equation is velocity equals the Vmax times the substrate concentration. And please note, in your lab manual, there is an error when giving this equation. They left off the S. So it's Vmax times S divided by Km plus S. So that's a very, very important equation that you're going to have to know for this experiment. And that's the Michaelis-Menten equation. So in this experiment, remember we're doing the lactate dehydrogenase reaction where pyruvate is reacting with NADH, making lactic acid plus the NAD. And then the NAD gets recycled in glycolysis. So the pyruvate comes from an endpoint of glycolysis, and I'll show you that in just a minute. Now, there are different pathways with which you can metabolize pyruvate. So normally in aerobic metabolism, you have pyruvate breaking down to acetyl-CoA and going into the citric acid cycle. But that's not the only thing that can possibly happen to pyruvate. There's different pathways for pyruvate in the cells. So this is just a picture from your basic biochem textbook. And so remember, you have, during glycolysis, you have glucose breaking down at the end to pyruvate. And then pyruvate has 3 different possible pathways depending on the type of cell and the availability of oxygen. So we've just talked about in the citric acid cycle that pyruvate can break down into carbon dioxide plus water through this oxidative phosphorylation process. But if you don't have enough substrates to be able to go through the citric acid cycle, there's sort of a shunted pathway that can handle any excess pyruvate and store it temporarily. And that's the lactate dehydrogenase turning our pyruvate into lactate through homolactic fermentation. And then as more substrates become available or needed in the citric acid cycle, the cells can take that lactate, turn it back into pyruvate, and then the pyruvate can go through the oxidative phosphorylation. Now, another thing, in other types of cells than humans, so this third pathway does not happen in people. It happens in yeast, though. And pyruvate can also be broken down into carbon dioxide and ethanol in an alcoholic fermentation process. So that's a third possible in some types of cells, something that can happen to pyruvate. So that's my favorite reaction right there. So remember, when you have an enzyme catalyzed reaction, and this is just a review of what we just talked about. You have the substrate going to product in the presence of the enzyme, and the enzyme actually is participating in the reaction because you have that enzyme substrate complex formed, and then after the enzyme substrate complex is formed and becomes product, the enzyme lets go of the product and is recycled back to pick up another molecule of the substrate so that the reaction can continue. So generally, what's happening in the reaction in this idea of steady state kinetics is that you have a very high concentration of substrate at the beginning, and you have no product at the very beginning of the reaction. And as soon as you add the enzyme to your substrate, which is going to include the presence of NADH as needed, the concentration of substrate starts to drop because the enzyme, the free enzyme that was there at the beginning of the reaction, now grabs some of that substrate, and as it grabs that substrate, it starts turning it into product. So the level of product starts going up as the level of substrates going down. Now please notice the concentration of the substrate is much, much higher than the concentration of the enzyme. If you have too much enzyme in the reaction, you're not going to be in steady state kinetics. It's going to grab all the substrate, turn it into product, and you're never going to be able to figure out a rate of a reaction using Michaelis-Menten kinetics. So Michaelis-Menten requires that you don't have an odd balance of substrate to enzyme. And that's one thing we're going to be checking in this experiment, making sure that we have enough substrate for the amount of enzyme that we are adding. So the free enzyme drops down low and becomes an enzyme substrate complex, and that amount stays steady throughout the reaction. So it's the total amount stays steady. But you'll remember from Chem 200 and Chem 201, when we're talking about something in equilibrium, we're not talking about something being the same being the exact same molecule all the time. We're simply talking about the same concentration, the same overall concentration of enzyme substrate complex. So what's happening is, one enzyme substrate is making product and grabbing another substrate immediately so that your concentration stays steady. You have a small amount of free enzyme. Most of your enzyme is in the enzyme substrate complex. The amount of substrates dropping, the amount of product is increasing, and these are all being done at relatively steady rates until you start to run out of a significant amount of your substrate. So generally, you want this reaction, your best numbers for the reaction are going to be before the reaction starts to slow, before you have used up 10% approximately of your substrate, you're going to get your most accurate numbers. When your reaction rate starts to slow, you are no longer in Michaelis-Menten kinetics, and so you should not be using any numbers that are not in a linear range. So let's talk about the actual basis for the experiment. And this might be a little hard for you to see, but this is basically NAD and NADH. So up here you have the reduced form of NADH, and here you have the oxidized form of NAD. And it's that difference in both of those that is making the monitoring of this reaction possible. Now, the AMP part of the NAD has an absorbance at 260. So for both forms, for NAD and NADH, you have very high absorbance at 260. So even though you might say, well, why aren't we using the peak absorbance for our NAD or our NADH? It's because while there is a small difference between the two forms at 260, it makes it very difficult to tell, which form it's in. On the other hand, if you're reading it at 340, there's a huge difference at 340. Because at 340, you have absorbance from NADH. It's not peak absorbance, but you have absorbance. And at 340 nanometers, the oxidized form of NAD has no absorbance whatsoever. So you're starting your reaction with NADH, which has absorbance at 340, and as the NADH is converted to NAD, you are losing absorbance. So it's a direct measurement of how much NADH is being converted into NAD, which makes a very nice assay possible. So what you're going to be doing in this enzyme assay. So the first thing you need to do is you need to say, well, how much time do we need for our enzyme assay to take place? Some enzymes happen some enzymatic reactions happen very quickly. Some enzymatic reactions happen very slowly. You can have enzymatic reactions that take place over, an hour, or you can have enzymatic reactions that take place in picoseconds. And picoseconds, you have to have special equipment to be able to measure it. Enzymatic reactions that take place in an hour are too long for purposes of this lab, which is why we've chosen a nice measurable reaction with a reasonable time course for this particular experiment. But you have to say, what amount of time do I need? So the first thing you're doing is you are mixing together your substrates with the enzyme. So you're mixing together the pyruvate plus the NADH, a certain amount of buffer to keep the pH steady. And you need to have buffer in there to keep the pH steady, because remember, the reaction is taking hydrogens to add across that carbonyl carbon. So if you did not have any buffer at all in the reaction, then you'd be changing the pH of your solution, and pretty soon you would not be in a good pH range for optimum pH to do your rate of reaction at all. So you have a little bit of buffer in there, and then mix in the enzyme and start measuring, how much product is being formed. In a perfect world, you directly measure how much product is being formed per time. And so there'd be an initial rate, and then after a while it starts slowing down, and by taking that initial product measurement, the change in absorbance versus change in time, that's your absorbance per time, and that can be converted with use of an extinction coefficient into a velocity for the reaction. So this is basically what you're going to be doing. And remember, I said that the initial rate's going to slow. 10% of your substrate is consumed. So when it starts to slow, you may have those data points on your graph, but you're not using those data points on your graph. But it's important to have them on your graph. So you have like an initial graph of product versus time, and I'm going to show you in a minute what you're actually going to be doing. This is a theoretical idea. Product versus time, and you have an initial rate, it starts to slow, and you say, okay, well, here's my whole graph, but I'm only going to use the ones that are linear. So that's how you know you're getting an accurate rate of reaction. So then you'd say, okay, well, here's the perfect time to use, so you'd want to make sure that your timeline is like this. So if your initial reaction took place over 6 minutes, but you saw that it was only linear for the first 3 minutes, for example, then all the future reactions can simply be done in 3 minutes, because you started off by saying, okay, we did it for 6 minutes, we saw that 3 minutes was enough, and now we're just going to do the rest for 3 minutes. So that's what you're actually going to be doing in part A of this experiment. So this is an actual example of what it might look like. So if you had an amount of enzyme added in. Now this is with a bunch of different amounts of enzyme. But let's say this was your part A curve right here, where you've added, let's say, 3 microliters of enzyme. So you add 3 microliters of enzyme, it starts to go down at a certain rate, and then you see it start to slow, and then you say, okay, you know, I've got enough data points to know that I'm no longer in a linear range. So the reason it's going down is you're measuring your substrates. The last graph I was showing you were just measuring some product, but we don't have the ability in this assay to measure product directly. Instead, we're measuring loss of the substrate, and our substrate, remember, is NADH. So our NADH has a high absorbance initially, around 2, 1.8, 1.7, whatever it starts off being, a high number, and it does not matter what your initial number is. From one assay to the next, as you get a tiny little, maybe bit more or bit less when you're pipetting of your NADH in the tube, your initial starting point may change. So the first reaction, you may have a starting number of 1.8, then it may go up to 2, then it may go down to 1.7. That's okay. What matters is the rate of the reaction, the change in absorbance with change of time. So you're saying, okay, I'm starting up here and I'm measuring the change in absorbance with change of time on this axis right here, and this is absorbance right here. And so this is your dAdt, which, using the extinction coefficient, is turned into the velocity of the reaction. Then in part B, you're going to be measuring by using greater and greater amounts of enzyme. And if you did not have any enzyme at all in there, the reaction would not take place. But as you add more and more enzyme, if you add then 4 microliters of enzyme, or 5 microliters of enzyme, or 6 microliters of enzyme, the rate's going to increase. And it's going to increase because you now have more enzyme in your reaction. So you have that same catalytic rate, but by increasing the concentration now of the enzyme, you now have the ability to make the reaction go faster. So what we're really measuring in part B of the reaction is we're making sure that we are in a linear range, that we have not yet added too much enzyme for the amount of substrate. So part B of the experiment, you're going to be increasing the amount of enzyme and doing that reaction all over again. So to make sure that you don't have excessive enzyme. Then you're going to graph velocity versus the enzyme concentration. So remember, you're going to use the extinction coefficient to turn the absorbance per unit time into a velocity. And graphing velocity versus enzyme concentration, with 3 microliters or 4 microliters or 5 microliters, you should see the velocity increase. Maybe by the time you add 6 microliters, now you start to get some curvature here. And that curvature is what you see when your substrate becomes limiting compared to the amount of enzyme that you have in the reaction. So then you'd say, okay, at 3 microliters of enzyme, I'm still well within the linear range of velocity versus enzyme concentration. I'm not approaching a point where my substrate becomes limiting. So then for the rest of it, you say, yeah, 3 microliters is a good amount of enzyme to use. One of the next things you're going to do is measure the effect of pH on the rate of a reaction. So you're going to be doing the same thing. You're going to be taking your substrate, your pyruvate, you're going to be taking your NADH, mixing them up with buffer, adding in the enzyme, and this time your buffers are going to be different pH buffers. So you're going to be measuring it at 6.5, 7, 7.5, 8, 8.5, etc. So a range of different pHs, and you're going to be measuring the velocity versus pH. So you should have a greater velocity at some pHs and a lower velocity at other pHs. Now, why that happens is pH can affect a number of things. So pH is able to affect the ionization of the substrate and the enzyme. So particularly in the catalytic site, if you have maybe a substrate that's differently ionized, it's not going to react with the reaction center in the enzyme properly. So as pH affects the ionization of the substrate and enzyme, maybe at a low pH it's protonated, or maybe it needs to be protonated at a higher pH, it's deprotonated, whatever. It can affect the ionization, and so if it's not occurring at optimal pH, you're not going to have a good rate of the reaction. It's able to affect the way that the substrate is actually binding, so not just maybe ionization attraction, but actually the way that substrate is able to bind into that reaction cavity of the enzyme. And it affects the catalytic activity of the enzyme, the ability of the enzyme to make the substrate into a product. And at extremes, it affects the actual structure of the enzyme. So as you get, say, in very high pH ranges or very, very low pH ranges, you can actually start denaturing or disrupting the enzyme structure itself. So the next thing that you're going to be doing in part D of the reaction is you're going to be investigating your Michaelis-Menten kinetics a little bit. So you're going to be measuring velocity versus substrate concentration. So you're actually going to be changing the substrate concentration while keeping the amount of NADH steady and while keeping the amount of enzyme steady. So you're not changing the enzyme, you're not changing the pH, now you're changing the actual substrate concentration. And so as you change the substrate concentration, your velocity can move higher. With higher and higher substrate concentrations, again, that's affecting that rate of the reaction to a certain maximum until that substrate drops out because you are at the absolute maximal velocity, that catalytic rate, which is just depending on the enzyme concentration, which we did back in part B, and so now we're on part D, where we're looking at the substrate concentration. So as your substrate concentration increases, what's happening in the tube where your reaction is happening, with more substrate, it makes it easier as soon as the enzyme lets go of the product and starts hunting around for its next substrate. With higher substrate concentrations, it's easier to find that next substrate molecule. So as soon as it lets go of the product, where's my substrate? Where's my substrate? Here you are. The shorter time that takes, the greater you can have your rate of reaction. But there's a limit. So you see that as you start increasing substrate, eventually it starts tailing off. And even though there's a theoretical maximal velocity, you can never really, in real world, reach that maximal velocity. And you can't reach the maximal velocity because as you keep adding greater and greater amounts of substrate towards infinity, the reaction itself would get thicker and thicker with that higher concentration of substrate to the point where the enzyme can't move around because you have so much substrate, so it's limiting the ability of the enzyme to move even though you theoretically have all this substrate in there. So you approach Vmax, but you never really reach Vmax. So how are you going to determine what Vmax really is? So there's something called a Lineweaver-Burke plot. And on this Lineweaver-Burke plot, you're plotting 1 over V versus 1 over S. So 1 over V versus 1 over S, and you are taking those same numbers that you got. You remember you just did that V versus S plot, and you started saying, okay, well, it's linear. It's not linear the whole way. But if you then plot 1 over V versus 1 over S, as you approach 1 over V, you're approaching infinite substrate concentrations. So this infinite substrate concentration is where 1 over V is 0, but you never really get there. But because you're extrapolating the line here, this intercept is 1 over the Vmax. So that's how you can find what your Vmax really is by extrapolating on a 1 over V versus 1 over S Lineweaver-Burke curve. And if you keep extrapolating in 1 over V versus 1 over S, the other intercept is minus 1 over the Km. So that's how you're going to find both your Vmax and your Km. So remember I said that the Km is your dissociation constant for the reaction. So if you have a very large Km, you're saying that the substrate has very low affinity for the enzyme. It wants to dissociate. So a large Km says it wants to dissociate. That substrate does not want to stick to the enzyme. On the other hand, if you have a very small Km, you have a small dissociation constant. So the substrate does not want to dissociate for the enzyme. So low Km, low need or want to dissociate from the enzyme, which means it has a high affinity for the enzyme. So both of these numbers are things that you can measure when you're trying to say, well, do we have maybe a potential inhibitor when you're trying to design a drug? Do we have an inhibitor for this reaction that we can use? You know, what binding constants do we want? You know, what Km, what Vmax? You know, can we make sure we're optimizing? And then they can tweak the molecular structure of the drug a little bit to try to potentially optimize. Okay, so the other thing you can do when working with inhibitors is start saying, well, what type of inhibition is it? So when you are thinking about inhibitors, you have different types of inhibitors. And the first type of inhibitor I'm going to talk about, and this is also in your lab manual, the first type of inhibitor is a competitive inhibitor. So the enzyme and the substrate are going to bind in the exact same place. So they're going to compete with each other. So here's a picture of what that would look like. So if you have a competitive inhibitor, so here you have the enzyme, and here you have your ideal substrate, and here you have the competitive inhibitor. So a competitive inhibitor is going to bind to the exact same spot on the enzyme that the substrate does. They're competing with each other for that same spot. So 3 types of inhibitors that I'm going to be talking about: competitive, uncompetitive, and mixed type or non-competitive enzyme inhibition. So back to this next slide. When you have a competitive inhibitor, this is what it looks like in like a reaction diagram. You can have the enzyme reacting with the substrate, forming an enzyme-substrate complex, which then goes on to make product. Or you can have an inhibitor bind to it. But if you have an inhibitor, substrate's not going to fit in there. If you have a substrate, inhibitor is not going to fit in there. So it's one or the other. Now you can overcome the effect of an inhibitor by increasing the amount of substrate. So when you are doing a graph where you're trying to determine what type of inhibition you're looking at with an inhibitor, you're going to potentially change the parameters of the graph. So as you add inhibitor, you're not going to be having potentially the same Vmax, or you're not potentially going to be having the same Km, for example. These are things which can change with inhibitors. So as you increase the amount of inhibitor, your V versus S graph would change, which means your 1 over V versus 1 over S graph would change. And so you'd have this line, this new line right here. And so this would be not the true Km and the true Vmax potentially, but Km sub I, for example, which we're not looking at. We're only interested in Km and Vmax for the original no inhibitor, but we're interested in what is changing when we add the inhibitor for purposes of this reaction. So as you increase the inhibitor, the line here is increasing because the rate of the reaction is potentially decreasing with the inhibitor. But as you move towards infinite substrate concentration with a fixed amount of inhibitor, you can potentially outcompete all that inhibitor. So you have a certain amount of inhibitor, but by increasing the substrate more and more and more, you can get to your same Vmax by increasing substrate. So when you are looking at the kinetics here, your 1 over Vmax stays the same, but your Km is changing because it's going to take more substrate to get to this same Vmax. So your apparent binding affinity of the substrate with the enzyme is what is changing, even though the maximal velocity is staying the same. So other types of inhibition, you can have uncompetitive inhibition, for example, and in uncompetitive inhibition, you have the enzyme and you can have the substrate binding, but in uncompetitive inhibition, the inhibitor is going to bind to a different place on the enzyme and stop the reaction when it binds there. So by potentially changing the shape of the enzyme or something, the substrate can still bind, but it can't go to product when there is inhibitor in there. So in an uncompetitive reaction, the inhibitor can only bind to the enzyme substrate complex. So the first thing that happens is a substrate binds to the enzyme, and the next thing that happens is an inhibitor can come in here when those two things are bound together, change the shape so that the reaction cannot go to completion. The other type, the mixed type or non-competitive, the inhibitor can bind to either the enzyme by itself or the enzyme substrate complex. So those are going to look a little bit different in terms of their reaction diagrams. So in uncompetitive inhibition, here we have, remember the inhibitor can only bind to the enzyme substrate complex. So we have the enzyme plus substrate going to ES, and then the ES can either go to the product or it can go to binding the inhibitor.